Bismillahirrahmanirrahim. Hasbunallahu wa ni'mal wakeel. Allah alone is sufficient for us and He is the best disposer of affairs. Now, coming to the paper number 33, which was just held in March 23, I'm going to be discussing the question 1, and this is as it is. The syllabus, if you remember, changed in 2022. So these are the few papers that we have for the new syllabus, which is of course not much of a change, but just a slight change, and you can see it in the questions. As you all know, the time is 2 hours, and this of course carries 40 marks. So the mark is 40 marks for this, and you must know that this is the only paper in which I have seen students getting 38 out of 40. So this is a very scoring paper. And you have one more exam on the first of this coming month, and I'm sure some of you will be taking that exam. So please make sure that you do your best and give it your best shot. Now, looking at the question number 1, you will investigate the effect of different temperatures on the permeability of the cell surface membrane of beetroot cells. Now think theoretically. So we are discussing the permeability of the cell surface membrane. So think, what is the structure of the cell surface membrane? It's a phospholipid bilayer, and it's got the phosphate head and the fatty acid tails, and they orient inwards, and these fatty acid tails actually prevent, and this is the hydrophobic part, and this is the hydrophilic part. This is the hydrophilic part, and that's the hydrophobic part. So just quickly revise it in your mind. Beetroot is a vegetable that contains a red pigment in its cells. When beetroot tissue is put into water, so if you take some water and you put the beetroot tissue in it, I'm using a purple color because it's a dark red color, which I'm not sure. If you put some beetroot tissue in water, the red pigment can move out of the cells. So the red pigment can move out of the cells through the cell membrane, changing the water to a red color. So the water color can change because the beetroot cells will be damaged when you cut it, and that will release the red pigment, and this red pigment will now enter the water, and you see this is the water, and so this is the water will enter and it will start to color it. You are provided with the materials in table 1.1. Number 1 is B, which is 2 pieces of beetroot, hazard none, volume none, W distal water, and this is none, and this is 100 cm cube. Now remember, beetroot and water, this will be a very low hazard, because as it says, the red pigment in beetroot cells can stain clothing and skin, so it cannot damage. It's not an irritant, it's not a chemical, but it can only stain your skin. So use blunt forceps to handle beetroot tissue, and if any pigment comes into contact with your skin, wash it off immediately under cold water. It is recommended that you wear suitable eye protection. You will need to put beetroot tissue in water at different temperatures. So we'll have to set up a water bath, and we'll have to have different temperatures, and we'll have to put the beetroot tissue in the different temperatures. And then you leave the beetroot tissue in the water for a period of time. The period of time might be specified as we read the question. And then we will record the intensity of the color in the water at each temperature. So first we'll take a test tube, then we'll take another test tube, we'll put the beetroot in this, we'll put the beetroot in this, and then something will happen to the water. So it will become either light red or medium red or dark red, because this is at 25 degrees Celsius and this is at 35 degrees Celsius. So the pigment is going to come out, and it is going to now stain this water. So record the intensity of the color in the water at each temperature. So the beetroot is in water. So there is the water here in which we place the beetroot, and the pigment is going to leak out, and that is going to color the water. Part 1 of the question, the temperature range that you will use must include a minimum temperature of 25 and a maximum of 65. So I always do it like this: 65 minus 25, this is 40, so let's divide it by 4 and we get 10. So state 3 other temperatures that you will use. So I would use 35 degrees Celsius, 45 degrees Celsius, and 55 degrees Celsius. So that makes it a very regular intervals of 10. 25 plus 10 is 35. 35 plus 10 is 45, 45 plus 10 is 55, and 55 plus 10 is 65. So we have 25 here, which was specified, and 65 here. So just writing these, because the units are given, you get 1 mark. Oh, it would be sad if somebody lost this mark. But I know students would have lost this mark. Now we start how to do the experiment. This sometimes, I was just I was just looking at the students who were doing the paper 3-3, and I was seeing how some students were struggling with all this. And it is sad, because that means what have you done in the 2 years in the labs? Now it says carry out step 1 to step 18. Now you may do it, you may not do it. You can just think of it and do it theoretically, but you must really think correctly. There is a skill which you must have in this. Step 1, cut the beetroot pieces in 2 mm thick discs. You will need at least 25 discs. So, okay, so this was the beetroot given to you. So you just cut it up into pieces, and you have to measure it. It should be 2 mm thick. So 2 mm thick disc, you will need at least 25 discs. Now put all the discs that you have cut into the beaker labeled D. So you have a beaker labeled D, and you put these pieces of beetroot in that, and there should be at least 25 pieces, and you cover it with 30 cm cube of distilled water, which is W, which is in the beaker W. So you cover it up with 30 centimeter. Now this beaker was labeled D, so there was a beaker labeled D already there lying on your place where you were doing this experiment. Then you stir with a glass rod. So then you stirred it. Now what do? Now you pour off this water. So you pour off the water into the beaker labeled for waste. So there was another beaker which was labeled waste. So you poured off this water here, leaving the discs in the beaker. So you finally left with the discs in the beaker labeled D. So the discs are now in the beaker labeled D. Now you have poured off the water. You have added that to the waste. You have now the discs in the beaker labeled D. Step 5. Put all the discs on a paper towel. So, okay, now all these discs go onto a paper towel. And you put all these discs on that, on a paper towel, sort of a tissue paper, and blot them to remove the excess water. And blot them. Blot them means you just tap them very softly so that you remove the excess water. So now you have all the discs on this blotting paper or this paper towel which we have. Step 6. Now set up and maintain a water bath at 25 degrees Celsius. So we've given you a beaker, you put some water in it, and you maintain this. So you have a thermometer in it, and you maintain this at 25 degrees Celsius using the beaker labeled water bath. So we've given you a beaker labeled water bath. The water bath will be needed in step 10. So now you've got this beaker, you've labeled it 25 degrees Celsius. Using the beaker, you haven't labeled it, sorry. So you set up and maintain a water bath at 25 using the beaker, and the water bath will be needed in step 10. Okay. Now you label 2 test tubes with the temperature of the water bath. So now, what do you have? Now you have 2 test tubes labeled 25 degrees Celsius, right? Label 2 test tubes with the temperature of the water bath. Put 5 discs into one. So you put 5 discs into one. 1, 2, 3, 4, 5 discs into one of the labeled test tubes. Put 10 centimeter of distilled water. 10 centimeter of distilled water into the test tube with the 5 discs. So you put this here. Put this test tube into the water bath. Now you take this test tube and put it in the water bath here. Put this test tube into the water bath for 4 minutes. You will need to maintain the water bath at the correct temperature throughout these 4 minutes. Right? So you have now left this. So you've taken the test tube, which you label 25 degrees Celsius, and then you put in 5 discs, and then you put it in the water bath for 4 minutes. After 4 minutes, remove the test tube with the discs from the water bath. Now you remove this, pour the water from it. So you pour the water from it in here, which means what? You leave the discs in this. After 4 minutes, remove the test tube with the disc from the water bath. Pour the water from this test tube into the second test tube that you labeled in step 7. So this was the second test tube, leaving the discs in the first test tube. So we are leaving the discs in this first test tube. So 2 test tubes labeled 25. You put 5 discs in it, added water to it, left it in the water bath for 4 minutes. Now after 4 minutes, you pour the water, because the water, what will have happened to the water? The water will have changed color. Because at 25 degrees Celsius, the partial permeability of the cell membrane would have been lost and the pigment would have escaped or leaked out into the distilled water. So the distilled water will not be clear. It will be a light red or a medium red or a pink red, because the pigment from the beetroot must have leaked out. Now put the test tube containing the water into a test tube rack. So now you're going to have a test tube here. And this test tube has 25 degrees Celsius written on it. So it has a certain shade of red in it. Then you will have other test tubes, 35, 45, 55, and 65. So you will have 5 test tubes here with different shades of red pigment which has leaked out because you have changed the temperature in each. And the temperature which you are changing is called the I am changing, that is called the independent variable. So you put the test tube containing the water into the test tube rack. Now put the test tube containing the discs. Now the disc, this test tube which had the disc, you put it in that beaker. And you put it in that beaker which was labeled D, which was empty at the moment, because what had you done is you had taken the discs out of it. So there's just a beaker there in which you would place the test tube containing the discs. Now set up the water bath at the next higher temperature stated in A2. So you see, remember, it was 25 degrees Celsius, then it was 35 degrees Celsius. And then it was 45, and then 55, and then 65. So set up the water bath at the next higher temperature in A1. So now you've done it at 25. Now you keep the water bath at 35. We'll give you some hot water, and then you maintain it at 35 degrees Celsius. And important thing, you repeat step 7 to step 15. So you do the same thing, 2 test tubes labeled 35, put 5 discs, add distilled water, a specific volume has been chosen, 10 centicube of distilled water, leave for 4 minutes, and then you pour off the water, and now this is for 35 degrees Celsius. So it said that repeat steps 7 to 20 until all the temperatures stated in A1 have been tested, finishing with a maximum temperature at 65 degrees Celsius. Step 17, observe the color intensity of the water in each of the test tubes in the test tube rack. Placing a piece of white paper or card behind each test tube may help with your observations. Most of all, letting you know what is actually beetroot. Maybe now that you've seen it, you can understand what is beetroot. So this is beetroot, and in Urdu it's called chukandar, and it's a very nice. You boil it and you have it in your salads. Now this is the sort of color that you would get test tubes after incubation in a water bath. The more opaque the solution, the greater its light absorbance. This suggests the cell membrane have been highly permeable and has allowed a lot of the colored pigment to leak out. Another diagram showing you this is that how this was at 20 degrees Celsius, so this is probably some other temperature, and how the color is different in different temperatures, because as the proteins get denatured, which are present in the phospholipid bilayer, more of the pigment will leak out. As you can see here, another very nice diagram is that you have left the beetroot at a temperature of 90 degrees Celsius, and you've left it for 10 minutes, and you observe the color. So what has happened? The vacuole containing the pigment, vacuole is surrounded by the tonoplast. So the vacuole containing the pigment leaks out of the damaged membrane. The damaged membrane, why is it damaged? Because at 90 degrees Celsius, all the proteins would be denatured. And of course, this will then, of course, result in the partial permeability being lost and will become fully permeable. So the cell membrane is damaged, the tonoplast is damaged, and all the pigment leaks out. Then step 18, using only the symbols shown in table 1.2 represent intensity of the color. Decide the intensity of color in each of the test tubes. In the test tube rack, record your results in A2. Now the intensity of the color, dark red shows you 6 pluses symbol, and then 5 plus, and then 4 plus, and then 3 plus, and then 2 plus, and then 1 plus. So no color is 1 plus. And so it is the decreasing, the darker the color, it will be 6 plus. Record your results in an appropriate table using only the symbols shown in table 1.2. You may use the same symbol for more than one test tube. So if you put, say, 3 pluses, you can put 3 pluses in another one as well. So that is actually immaterial. They know that there can't be much of a difference in these sort of temperatures. Now here comes the most important thing. You record your results in an appropriate table, and this is for 5 marks. So these 5 marks are very important because you can get full marks here. But if you are very careful in drawing the table. Now, I will just give you the main points of the table. The first thing is the independent variable is always on in the first column. So the first column is going to be the independent variable, which is what? Which is what you changed, and that was temperature. So temperature, write the whole thing. Sometimes they don't like you. So degrees Celsius, you write the units here. We had 25, 35, 45, 55, and we had 65. So then you draw the lines. I'm not very good at that. Some of you are brilliant at that. My drawing is a little crooked. Now, what is the other thing which you're going to be talking about? Now you can see is that this is what is mainly asked and was required. So you've given me temperature in degrees Celsius, you've given me the intensity of color, and you've written symbols in it. Now you record the data using symbols for all the temperatures. Results for all the temperatures, records using symbols and the correct trend. So the 5 marks are very easy to get. So, well, I would say this would be 1 plus, 2 plus. No, I could not do the experiment and do this. I might do just one of them at 25 degrees and one at 65, and then just make up the result. Because you see, there's no way that it has to be checked, and it can be very different for different students. So it's not necessary that it's going to be. So somebody giving me this result is fine. Because the higher the temperature, theoretically, when you look at it, higher the temperatures, more the proteins will be denatured. So more the pigment will leak out. So it will be a darker red color. Now, what is the intensity? What does it say? Dark red is 6 plus. No color is 1 plus. So similarly, this is what I have done is so it is not, it's not a very difficult thing to do. You can always just cook up the results. It doesn't matter. It's actually theoretically there's only 1 mark for this for the correct trend, and there is not going to be compared with every student is going to have a slightly different version of it. Somebody might have 2 plus here, and then maybe another 2 plus here, and then maybe 3 plus here, and then maybe 4 here or 5 here, and then 6 here. So I mean, even this person's is correct. So what you have to understand is just be confident and just do the paper. You've got to think of it theoretically. What is the theory behind it? Now coming to the part 3 of the question, state the independent variable in this experiment. Everybody, I've just talked about it, is temperature. This is what you have changed. Independent variable is what you change. So independent variable was temperature. Then it says use your knowledge of cell surface membranes to explain the results that you recorded in A2. Okay, why has the color become darker as the temperature is increased? Now what is going to happen is we're going to talk about the phospholipid bilayer. We're going to be talking about the proteins being denatured, and we're going to say this increases the fluidity of the membrane because it's for 3 marks. So increases the fluidity of the membrane. That means all of the things becomes totally permeable. You see, it's partially permeable. So increases the fluidity of the membrane, and that was the 3 marks. So that was a difficult 3 marks, but I'm sure many of you got that 3 marks. So phospholipid bilayer proteins denatured, increased fluidity of the membrane. Step 2 to step 4, improve the validity of the results by removing the pigment that was released when the beetroot tissue was cut. So initially when you put the beetroot in water, that is what we're talking about. State how you could confirm that all the pigment released when the beetroot tissue was cut has been removed. Very simple. When you take the beaker and you put the beetroot in it and then you add the water, so this water should come out clear. That means no more pigment is coming out anymore. So water has no red color, or water has become clear. So water has no red color. How you'd confirm that all the pigment when the beetroot tissue was cut has been removed? The water has no red color. So that was the basic thing which you had to write for that. Then it says two significant sources of error in this investigation. Two significant sources of error in this investigation are shown in table 1.3. Complete table 1.3 to suggest how to make an improvement to reduce each of the sources of error identified. How to make an improvement? Difficult to judge the intensity of the color. That was very easy. Use a colorimeter. You use a colorimeter. Colorimeter is the one which we talked about a lot of times in a paper 5 as well. So colorimeter is the one which we are going to use. Then it says error was difficult to maintain the temperature of the water bath. So you use a thermostatically controlled water bath. And I always say that whenever you're mentioning a water bath in every paper, whether it's in a paper 2 or anything, you always say use a thermostatically controlled water bath. And that is for you to, because they leave these sort of 1 or 2 easy points for every student to get this mark. So use a thermostatically controlled water bath. Next comes the graph. A scientist investigated changes the mean width of stomata in the leaves of a plant growing in hot, dry conditions. The scientist measured the widths of stomata at different times of the day. From 200 hours to 2200 hours shows where the scientist measured the width of each stomata, width of the stoma, guard cell. The scientist calculated the mean width of stomata for each time of the day. The results are shown in table 1.4. So this is a table given to you. Now this is always, the first column is always the independent variable, and this always goes on the x-axis. I say this time and time again, and if anybody makes this mistake, it makes me very, very angry. Very angry. My Pathan DNA just boils. And so time of the day in hours: 200, 400, 700, 1500, 2200. Mean width of the stomata in arbitrary units. So the smallest unit is 2 and the biggest unit is 795. Here we have 2 and 22. So now you've got to figure this out. What scale are you going to take on the different axes? Plot a graph of the data shown in table 1.4 on the graded figure. Use a sharp pencil. Just labeling the axis correctly and fully, and not missing out any words, and not shortening anything, and not abbreviating anything. Now just doing this gets you 1 mark. When you look at it on the x-axis, you had figures from 2 to 22. So if you look at it 2 to 22, I wrote that again and then I wrote it again, and so I must be getting 22 somewhere here or at the end. So I took a scale of 4, 8, 12, 16, 20, 24. This would help me. The 22 would somewhere come in between. Well, you could have taken that. You could have even taken 5. That was also allowed in the mark scheme. Taken on the y-axis was 20, 40, 60, 80, 100. On this, it was this. And of course, then you have to just plot the graph and then connect the points with a ruler. So this is how I plotted the points, and either you plot them with a dot and put a circle around it, or you make a cross. I'm more comfortable with a dot and the circle. It has to be a dot. It can't be a blob. You can't be like this. It has to be a dot, and you have to circle it. So you connected the points. Now, many students ask me, how do you connect the points? I say connect the points with a ruler, point to point with a ruler. In this situation, this was a very one in which you needed to do that. You can also do a curve through it, but still you can make a mistake in the curve. So it's better if you just join point to point with a ruler. Is number two, use your graph to estimate the mean width of stomata at 300 hours and show on your graph how you estimated your answer, and this is for 2 marks. Now this is of course another thing which you should not be making any mistakes on it. So then you have to look it up, where is 300 hours, and then of course you take from the graph and you show it on the graph. So then you locate the 300 hours, because this is the hours part, and then you draw a line and then you read off this. Now this is somewhere between, it's a bit less than 70, or maybe 69, 68 or 69 or 70. And this is of course what you do from the graph, and you would write that on the answer. But you see, what are the 2 marks for? The 2 marks are not for this answer, and the units are already given. The other mark is for you showing it to me, showing this line for me on the graph. Did you show this line on the graph? If you did, only then you get the second mark. Coming to the last part of the question, which was the best part of the question, and I like that part very much, is because it really challenges the student who's really diligent in their work. With reference to table 1.4 and figure 1.2, describe the change in mean width of stomata between 200 hours and 2200 hours. So you had to refer to what? The figure 1.2 and the table 2.2. Now, this is table 1.4 and figure 1.2. So this is table 1.4 and this is figure 1.2. You have to refer to these. Now, what is the thing? And is that I have told you a million times, I think, in my videos, is what is a describe? Describe is no biology. So between 2 and 700 hours, between 2 and 7, what has happened? Between 2 and 7, width of the stomata decreases from 86 AU to 4 AU. So from 86 AU, it has gone down to 4 AU. From 86, it has gone down to 4 AU. No bio, please. Don't give me the reasons for it. Describe means no biology. So at 86, it's gone to 4 AU. Then between 2 and 15, between 200 hours and 1500 hours here, this time, between 2 and 1500 hours, the width of the stomata decreases from 86 to 2 AU. So here it has gone down to 2 AU, arbitrary units. And then between 15 and 2200 hours, the width of the stomata increases from 2 arbitrary units, it increases to 95 arbitrary units. So describe is no biology, and you had to just give me these figures, and you would got your 2 out of 2. So 3 points. Between 2 and 7, width of stomata from 86 AU to 4 AU. You could have either said this, or you could have said this. Between 2 and 15, width of stomata 86 to 2 AU. Between 15 and 22, width of stomata increases from 2 to 95 AU. So you're giving me the decrease and the increase. That is the most important thing. When you describe, you've got to give me the increase. You have to give me, okay, what is the general increase, sharp increase, gradual increase, and then you gave me the increase. So decrease and increase, or you could have even given me the plateau. So that's why the decrease has been allowed between 86 to 4 or between 86 and 2, because that was a decrease. Or you could have said an increase from 2 to 95. So 2 marks and 2 points which you had to give me for the describe. So everybody could have got those 2 marks. That completes question number 1, and thank you very much. I will be continuing with the question 2 in the next video.